And now we're going to spend a little bit of time talking about antibodies and iCn3D. Okay, and I'm going to remember to share my screen in just a second here once I find the beginning.
While Sandy's doing that, we didn't build breaks in, so feel free to take breaks as you need, and that's part of why we're recording things.
Yep. I mean, I can just talk a lot faster, but yeah, we want to make sure that we hear our speakers and we have time to get going and start working. Okay, so I'm just going to give a really brief overview and introduction to antibodies and iCn3D. Many of you have some familiarity with antibodies, but they are proteins. They are made by vertebrate animals. I meant to put that in and forgot to say it, vertebrates. And they have 4 chains, a molecular weight of about 150 kilodaltons and altogether. And the chains are, there's 2 that are light. We call them light because they are. they weigh less. Okay, 2 are lighter. And those are, we call light chains. And then we have 2 that are heavier, and we call those heavy chains because those are 50 kilodaltons each. The light chains are about half the size. The antibodies have a business end here that binds to antigen, interacts with antigen, and they have heavy chains. And early on, when I first took immunology, I learned a lot about protease. I learned that people studied antibodies with proteases. They used things like papain from pineapples to chop their antibodies into fragments, and they looked at what the different fragments did. And that's where a lot of the names come from that you hear when we talk about antibodies. So these fragments bind to antigen, and we call them Fabs. And then we have this other fragment that was called the FC region because it would crystallize. As far as function, as I said, these are the ends that bind to antigen. This end, the FC region, is recognized by immune cells and by immune proteins like complement proteins, and it can stimulate a whole bunch of other kinds of things to happen. When you look at molecular structure models, you don't typically see many intact antibodies that have the whole entire heavy chain, light chain setup. A lot of what you see are these Fabs, smaller versions. Antibodies also have a hinge region, and there's more disulfide bonds than I put here, but those disulfide bonds hold the antibodies together, which is why when we run antibodies on protein gels, we can treat these with denaturing agents like DTT, dithiothriotol or beta-mercaptoethanol, and break the chains apart. And we'll see these at the 50 kilodalton point, and these ones at the 25 kilodalton point when we run them on a protein gel. Okay, humans have 5 kinds of antibodies that you may have heard of. B cells have IgD on their surface. IgM is one that's made, the first thing that gets made in response to an infection, and that's that really big one that I have a link to a cryo-EM structure in the Slack channel. IgG is the one you see most of the time and you hear about, and that one's the one that we find in serum and milk, crosses the placenta, and it can bind really strong. She'll come in, I'll explain a little bit about that later. And then we have IgA, which can also have a high affinity. That's the antibody that you would have inside your nose or your throat or on other kinds of mucosal surfaces. And it can be found as a dimer form, which I have a link to an IgA structure too. And then we have IgE, which are associated with allergies. Okay. Antibodies have variable and constant regions. V for variable, C for constant, and this is just a generic IgG here. And you can see the variable regions are the end that bind antigen, and the constant regions are the rest of it. And this is really cool because you think, okay, how do we have enough antibodies so that we can respond to challenges to our immune system that we encounter in the environment? Because we encounter lots of things that our body wants to protect us from. And that's because we have lots and lots of antibody genes that get recombined in interesting ways. This picture is from, well, Todd drew this picture actually and posted it on our Digital World Biology website. And I have a link to it in the background reading. From the BCR is another receptor blog. And what you can kind of see here, actually, I wonder, can I make that bigger? Yeah, I guess I can. Cool. A little bit. All right. What you see here is with, this stuff, we're going to ignore the T cell receptor, but antibodies, we have 3 loci that are coding for antibodies, 1 for the heavy chain, 2 for light chains. There are 2 kinds of light chains, kappa and lambda. And we have antibody genes are put together from a lot of different genes. The heavy chain here, we've got 38 to 46 different V genes. We've got 27 different D genes. Those are short, like 10 to 30 nucleotides long each. And we've got these J genes, 45 to 55 nucleotides, only 6 of those. And then the constant regions, those are the ones that determine the class of antibody. If it's IgG, IgM, IgA, IgD, I think I have them all. There's some different forms of IgG as well.
Sandy.
Yeah.
The graphic you're showing, this is regions on the protein, not genes, right?
No, these are genes.
Thank you.
Yep. These are on chromosomes. This is on the heavy chain one is on chromosome 14. And, you know, I don't remember, Todd, are kappa and lambda, where are they?
In terms of chromosomes?
Yeah, what chromosomes are they on?
2 and 22.
Oh, of course. Like everybody knows that.
It's on the right side of the figure.
Oh, that's helpful.
Yeah, yeah. Yeah, I don't remember that stuff.
Yeah, well, Todd worked in a lab that was sequencing a lot of these kinds of things. Didn't you help sequence one of these TCR receptor, T cell receptors?
No, I helped to annotate it, TCR alpha. Yeah. By then I had moved from the bench to software. So people would hand me their data and say, could you take care of this for me?
Yeah, and I have to admit, my students were always confused because I'd say, look, there's 38 to 46 different V genes. And they're like, but doesn't each gene have its own promoter and regulatory sequences and stuff like that? And in fact, no, these are just the protein coding segments.
Yeah, gene segment would be a better name, but it has the unfortunate name of gene.
Yep, they were named before anybody had those kinds of debates.
Now, one more, the BCR and TCR here, these are the targets of the antibodies, right? Because TCR is not an antibody itself.
It's not. I should have chopped it out of the figure.
Yeah, yeah. So the figure is the human antigen receptor loci. So as we've gotten into things like immune profiling and things like that, people have, they got tired of saying antibody and T cell receptor. So we needed a 3-letter acronym for antibody. So we call them B cell receptors because they come from B cells. And T cell receptors come from T cells. And that's why the title of the blog is BCR is the new antibody, T cell receptor is still TCR. Yeah.
Cool, because there's a protein BCR that plays a role in BCR-able.
Yes.
Yeah.
Thank you, Todd. That helps.
Yeah. Okay. Well, anyway, antibody genes, the final form of an antibody gene, is created by choosing, well, by a recombination event that takes place, and this is more clear in the blog, between a V gene, a D gene, a J gene, and the constant region. And in fact, you can switch from one constant region to another as a cell goes through its developmental stages. So V is joined to a D and is joined to a J. It's kind of neat, VDJ. And that is happening. And right, as Francis points out, the first antibody that we make is a cell surface receptor, IgD. So that happens with IgH. We join V to D to J. With kappa and lambda, we don't have a D region. We just join a V region to a J region. And this allows us to make a lot of different kinds of antibodies. Now, I've only put the functional numbers here, and I put the minimum combinatorial diversity here, and I put the link to where I got this information, the immune genetics database. But you can see if we take 38 different IgH segments and we combine, we have 38 times 23 possible junctions times 6 now possible junctions, we can have up to 5,244 different IgH variable regions, okay? And then we can get 170 different kappas and 116 different lambdas, but that's not all. Because when the recombination happens, the enzymes are sloppy and they can add bases. So we can get like up to 20 more amino acids added between the different segments. And afterwards, if it's IgG, we can even have a process called affinity maturation where it can get additional mutations and even stronger, which is why booster shots are a good thing. Okay, because your later antibodies are going to be better, at least to some forms of a virus. Okay, different forms of antibodies. Okay, here, remember we have the variable regions. Here we've got the constant regions. We talked about IgG. We talked a little bit about the Fabs, which are just the regions that bind to antibody or bind to the antigen. There are some other versions that people have made where they take the variable region from a heavy chain and they have a linker molecule that a linker sequence that they add with a lot of glycines and serines that can join these two. So we get a single chain FV antibody. FV is the fragment with the variable part. Okay, and then we can have nanobodies, which are a single domain antibody. And those can be generated from antibodies from camels and sharks and llamas. They're really of interest now because they're small. They're only 15 kilodaltons. So people think they have a really interesting potential. Okay, iCn3D. I'm going to now, I'm going to take that out of there and I'm going to go to iCn3D and we're going to take a look at iCn3D.
And everyone, if you want to take a look at the chat, because there's some clarification on this nomenclature and how antibodies go through their process. So,
okay, so we're going to look at IgG to start. And I'm at the NCBI right now, which is a nice place to start with looking at antibodies because you can search for structures here and you can learn about them. You can see this one, immunoglobulin, comes from a mouse, Mus musculus. Eventually you start to learn the Latin names for a lot of different organisms. But I could read the abstract for it. I could see the resolution of the structure. And I can see how things are arranged, and I can scroll down and I can see that there are the different names of the chains, and I can see there's lots of sugars in this molecule as well, in this antibody. And then I'm going to open it up and we'll take a look. Okay, good. Make sure it opens in the right place. All right, so now we have IgG. I mentioned that there are a lot of sugars, and you can see the sugars down here at the in the FC region. And each one is named here, and you can see how many there are. And I could also kind of scroll over them and see what they look like. There are, as I said, there are 2 heavy chains, and we can see that because they're longer. And there's the other heavy chain. So 1 green, 1 blue, and 2 light chains. And I can see they're light chains because they're shorter. I'm going to make my window just a little bit bigger here. By the way, these little corners, you can use those to drag the windows and make windows bigger. There is also a key on the right-hand side, and I can use the key to identify the different parts. You can see here that the blue rectangle is longer than the pink rectangle. So I know that's a heavy chain because it's bigger. Again, another heavy chain, light chain. And in fact, I can, sometimes you'll even see the names up on top. It'll say heavy chain, light chain. Okay, so now what I'm going to do is show you how we can look at some of the different domains. Here, this says IGV. That's the variable region of this chain. And if I just click it, you can see it got highlighted. If I click the constant region, that gets highlighted. And that will work with all of these. You can see what the different things are. How did they determine the structure if the sequences used are also different? Great question. Actually, what they did is they, this is a monoclonal antibody, which means that they cloned the genes from a B cell that was just, it was fused with a myeloma cell, and it was just making this antibody. So from this, they could purify the protein, and then they could crystallize it and get the structure from that. That's a very short answer, but you know, most of it.
The key being that each B cell produces a unique antibody. And so if you can purify those B cells in some way to separate them from one another, then you can get the gene of the fully assembled gene of an antibody, a unique antibody.
Okay, right. All right. Other things about iCn3D. I can, you can see I've got this selected. I can clear the selection by opening the select menu, and I can say clear selection. And I can actually get rid of the label too. There's another toolbar with some other options here, and I can say remove labels. Maybe I'll just leave that toolbar there for now. I can also turn my structure around. If I'm using a touchpad, I can, or a mouse, I can click and drag to turn my structure. I can zoom in and out by using 2 fingers to drag away from my computer. And I can push my fingers towards the computer and zoom in. Or there are also keyboard commands that I can use. I could use Z to zoom. And then right next to Z is X, and it goes the other way. So if you're having troubles with using your fingers, then you can use that. I want to point out too that I put a quote into my notes that all models are wrong, but some are useful, from George Box. Thanks, Todd. And this is to remind me that we're not looking at an actual antibody here. We are looking at a model of an antibody that CN3D derives and displays based on the experimental data that it has in the structure file. So the experimental data that the researchers provided. And you see there are some breaks here in the chains, and I'm interpreting that to mean that CN3D might not have the data to display the structure in those areas. Okay, so we have the chains, we have the domains. Now there's a cool thing I like to do here with looking at orientation. Okay, I'm going to choose rainbow, and I'm going to click chains. And what I've just done, I'm going to zoom out a little bit. What I've just done is color the protein chains from the amino end to the carboxyl end. And this goes in the same order as the rainbow. Roy G. Biv ROY-G-BIV, right? Red, orange, yellow, blue, green, blue, indigo, violet. And what's cool, if I look at the sequence and annotations window and I click details, I can see that my sequence here is colored the same way. Okay, it's colored the same way as the structure. The carboxyl end of each protein sequence is going to be violet, and the amino end is going to be red. Okay, we talked about domains. I think I'm going to skip disulfide bonds right now, but show you quickly a little bit about binding to an antigen, because Erin's going to come at 9:30 and I don't want to take up her time. So I'm going to find another antibody for you and show you some antigen binding. So I'm opening up file, saying retrieve by ID. MMB ID is going to take me to the, it's going to allow me to put in an ID from the NCBI. Now, I never remember MMDB IDs, but I can usually remember PDB IDs, which are only 4 characters, much easier to type. 6xDG is an example of a structure that contains 2 antibodies bound to the receptor binding domain from SARS-CoV-2. Okay. Now, which is which, you might ask? I'm going to open up the analysis menu and choose sequences and annotations. And that gives me a key that I can use to tell which is which. I can see right away here that this is the spike protein, is the pink structure, the pink molecule that's in here. And these guys are Fabs. And I can tell right away that these 2 different Fabs are binding to different sites on the spike protein. So the spike protein is an antigen, something that stimulated an immune response. The different sites that the antibodies are binding to are different epitopes. So each site that an antibody interacts with is an epitope. Okay. Yeah, all right, interesting. Amy's commenting that there can be errors when you use the PDB only option versus the MMDB only option. And cool, if you type in a PDB code in the MMDB spot, you maybe have less errors. I guess that's what I always do, but I never think.
Because you're getting the MMDB file, it's
Right.
referenced with the PDB ID.
Yes.
Yes, we had cofactors missing, like the NAGs were missing when we tried to type in the PDB code in the PDB only spot, not the one that comes up at the beginning, just so you know.
Yeah, definitely good to know. All right. I'm just kind of scrolling through. Here's who's here for a minute, because I know Erin should be here any minute. All right, so how do I look at interactions between antibodies and their epitopes and identify what the epitopes are? What I do is I use this region right here as a key, and I go to select, and I can say select by distance, because if something is interacting with something else, they're going to be close. All right, I'm going to be closer. So let's see. And if everybody could mute themselves, that would be helpful, I think. There's probably a way I can do it too, but not very easy. Okay, great. So, so what do I want to do? I'm going to pick the light chain here, just from this antibody, 10933. And I scroll through here to find it. So here it is, 6xDGD. And I'm going to see what it interacts with on the SARS-CoV-2 receptor here, receptor binding domain. That is going to be 6xDGE. All right, so I've got them interacting and I say display. And if I turn this, I can see that there's one, looks like there's maybe one residue of aspartic acid, or let's see. I can zoom out a little bit. Yeah, maybe asparagine. I can sometimes zooming in helps with that. Yeah, okay, phenylalanine 486. I'm also, what I'm going to do is I can save what I've just selected so that I can work with it later. This is really, really helpful, especially if you're going to be selecting and deselecting things. So I'm going to say, I'm just going to call this the light chain epitope. Assuming I can type 10933 and say save. Now you see that it's going to appear in my list of things, which is great. I can also, if I go to details, I can see which amino acid is highlighted here. There we go. And I see it's an F for phenylalanine. And I can see if I hold my cursor very carefully, it's really tiny. And I go, no, you guys can't see this very well probably, but it says F486. So it's phenylalanine 486. All right, so that's what the light chain from that antibody is binding to. I can do the same thing with the heavy chain. So I'm going to go back to my key so I can see what the heavy chain is. And I see the heavy chain is this brown sequence, 6xDGB. So I'm going to go to by distance. I'm going to select the heavy chain. I want to see the heavy chain interacting with the receptor binding domain. So I'm picking that. I'm going to say display. And you can't see it because it's behind there, but you can see there's a lot more amino acids that have been highlighted, which is cool. So the heavy chain binds to a lot more things than the light chain. And I can take this and I can say save selection and give it a name. So I'm going to call this heavy chain epitope. Say save. Now, if I want to look at any of these, I can just click them in my list here and see them. I can also look in the sequence and I can see, I go to heavy chain epitope, should appear here in the sequence. And I can see all the amino acids that the heavy chain interacts with. All right, and what I can do too is I can save my work. So I'm going to take this and I'm going to say save file, share link. I could save a file too, but I'm going to make a link and I'm going to post this link into Slack and you guys can look at it later on. Okay, and then I'm going to stop for now and see if Erin is here. I think I'll post
I
this.
am.
You are. Great.
I am.
Well, thank
Hello.
you. I sure appreciate you being here, Erin, because Erin is one of the experts and one of the, probably somebody who has more expertise with cures and undergraduate research than anybody else I know. And Erin, I'm going to let you introduce yourself beyond that.
Absolutely. Hi there. I'm Erin Dolan. Shall I just go ahead and launch in? Do y'all want to like take a stretch break? I don't know how long you've been going.
Or do you want to stop the video so you have a clear start when cure starts?
Yes, I